Here you go. Thank you. Well, I'm Steve Shapiro. Maybe a little different. I'm a medical geneticist. I have an affiliation with MUSC. Interestingly, it's in the College of Dental Medicine. That's a long story, but I'm in private practice, and I see a number of patients with Ehlers Danlos, and I was asked to give this talk today. Interestingly, I learned about this talk from one of my patients last week, so didn't know much about this conference, didn't know much about who was coming, so I've kind of done a talk at a certain level that hopefully won't bore the professionals in the office and it's not over the head of the lay people in this group. I thought it'd be important to understand why we think Ehlers Danlos or the Ehlers Danlos syndromes are genetic or inherited. And so I've set up a series of questions. One question is Ehlers Danlos syndrome? And the word syndrome comes from the Greek, which means running together. So what we see are symptoms that are recognizable, and a collection of symptoms and physical findings. Many times the syndrome, we don't have an etiology. In this instance, we do. So the answer to the first question is yes. Is Ehlers Danlos a syndrome? And depending on who you believe, there are 12 or 13 different types of Ehlers Danlos. And here's a list of the 12. Many of you are familiar with the very classic EDS, the original reports by Drs. Ehlers and Dr. Danlos that talked about very hypermobile people with very stretchy skin, and that's what we call type 1 or classic. Most of my patients worry about the vascular type, which is fairly rare. But and again, the most common obviously is the hypermobile EDS, and unfortunately we don't have a genetic marker for it as of yet. So the second question, is Ehlers Danlos inherited? And when we look at inheritance, we see there are three basic types. There's Mendelian, chromosomal, and multifactorial. Well, Mendelian is named after Gregor Mendel, a monk in Austria, and who did these very famous experiments for peas. And he was the one that coined the dominant and recessive inheritance. People have looked at his data, and it is so perfect that people believe that he really cheated on it. So he may have been the first person to cheat in science, at least in genetics. And so when we look at Mendelian inheritance, we know that there are these different types. There's autosomal dominant, recessive, X-linked, recessive, X-linked dominant, and Y-linked. The word autosomal means that it is on an autosome. Remember, in your biology, as a human being, we have 46 chromosomes. We pair them together. 22 are what we call autosomes. There's a set that we call sex chromosomes. And if you're a female, you have 2 X's. If you're a male, you're an X and a Y. So there is inheritance in all of those genes. The Y chromosome doesn't have very much. It confers us to be males and maybe hairy earlobes. Now, and I think one of the more interesting things is nature loves symmetry, and so how do men exist when we don't have as much genetic material as women? My wife would say that women are superior because of that, and it's hard for me to argue that point. But there is something called lionization, named after a Mary Lion, and her research is that there's a random inactivation of an X chromosome, so that women basically have the same chromosomal material as men. And that's why sometimes on X-linked recessive disorders like hemophilia, women can be affected because they can have the bad gene turned on their X chromosome more than the normal one. So how do we know something is dominant or recessive? Well, again, one of the first steps in genetics is to do a family tree, to look at thousands of families and to get an idea about, can this be inherited? And when we look at dominant, we see that males and females are equally affected. It only takes one allele. Remember, we get a set of genes from our mother and a set of genes from our father. If you have an abnormal one and it's expressed, it's a dominant. And again, the basic genetics, and I won't bring out the Punnett square for all of you, but 50% of your children, if you're affected, can be affected. The trait tends to be in successive generations because of the high risk of 50%, and that's what we call vertical transmission. One of the most important things is that there can be male-to-male transmission, meaning it's not an X-linked disorder, because men can only give their wives to have boys. And last, individuals who are unaffected can't have children who are affected unless under a very unusual situation. So what are the other basic assumptions and dominants? And I think this really goes to Ehlers Danlos. The basic defect involves structural proteins. And Dr. Maitland has talked about a little bit about that. She almost gave all of my lecture away, but which is great. Sometimes you can get, if you have one gene, one abnormal gene tends to be less severe than if you have two abnormal genes, although it's very rare to happen, but it does occur. There's something called penetrance. That is, you can have a gene and never have it really express itself. So sometimes we see people who have parents who are affected, they're not affected, but then they have children who are affected, and that would be incomplete penetrance. I think that the most important thing in dominant is there's a variability of expression. That's why no two of you are alike in terms of your clinical manifestations. And that's really important and makes it very difficult for people to make the diagnosis. You can have a list of 50 different symptoms in a disorder. No one has all 50. And everyone can be different. And remember, you only share 50% of your genes in common with your kids or your parents, and that admixture could really add into the variable of expression. We see spontaneous mutations are common, especially when you have an older father. This is one of those things that drives me crazy in the 2017 international guidelines. One of those things is, do you have a positive family history? Well, if you're the new mutation, you're not going to have a positive mutation. So, and anyone who knows me, I'm not a big fan of the 2017 guidelines, and I'm hopeful there'll be some of us will rise up and try to get better diagnostic criteria for hypermobile EDS. Males and females are equally affected, and as you can see, here's the list of the EDSs that are dominant. And again, the hypermobile is, by our pedigree analysis, an autosomal dominant. Now, there are recessives in this world, and recessives, again, autosomal, meaning men and females are equally affected. You must have two abnormal alleles to be affected. Generally, your parents are unaffected, but are carriers, and carriers tend not to be affected. And again, this is where people say it skipped a generation. It really, the gene is there. It just didn't meet its pair to have to be affected. And again, if you've had a child with a recessive disorder, there's a 1 in 4 chance to have another child with this particular disorder. And again, some of the assumptions, the basic defect tends to be enzymes, or enzymes that produce proteins. It's usually an all or none, so penetrance is not as different as in when we see recessives. The expression tends to be consistent. And again, consanguinity is a term maybe you don't know. It's marriage between blood relatives. So that's the reason why in the Bible it says you're not supposed to marry your first cousin, because you're more likely to have the same rare recessive gene in your family and therefore be at higher risk. What they also don't say is you're more likely to have some genius genes. And so we see both ends of the spectrum with recessive. And so consanguinity has its pluses and its minuses. And again, the risk is low unless you marry one of your relatives, or there's a high frequency in the population. Something like sickle cell in the African American population, 1 in 20, or cystic fibrosis, 1 in 20 in the Caucasian population. You're more likely to meet that one than some of these very rare ones that we see every once in a while. And again, we have in the 12 of the EDSs, we have a number of the recessives, and they're very, very rare. And as you can see, you know, they just are not all that common in the population. So the next level is we do know that EDSs are inherited. They're syndromes and they're inherited. Now can we find the genes? Well, the first genes that were found were for classic EDS in the 50s, the COLA 5A1 and the COLA 5A2 gene. And then in the 80s, we started having a really rapid discovery of the genes. And if you'll give me a moment, I always want to plug the great state of South Carolina. We don't get a lot of credit here a lot of times, but one of the techniques that really made DNA discovery possible was done by a guy named Kerry Mullis. He went to Dreher High School in Columbia, South Carolina, and he was the one that found PCR, or he won the Nobel Prize for it. PCR, as for those who don't know, is the concept that you can duplicate DNA. He found out, or he put together the fact that if you put DNA in hot water, it opens up. If you put it in cold water, it closes. If you put nucleotides in, you can grow DNA. So you can take a small bit of DNA and get a lot of DNA. And with that, with those quantities, we can really do research in it. So he's one of 5 South Carolinians that won a Nobel Prize. Interestingly, he won it for chemistry and not for medicine, but hey, we'll take it any way we can in the great state of South Carolina. So so the discoveries in the 80s led to finding most of the genes for EDS, and frankly for most of those. And so, but before that, even before that, we used to use a system called linkage. And that's the concept that genes tend to be together. And if we can identify another gene that runs together, then we're likely to be able to find it. And the problem with this is we need lots of families and large families to do it. We use something called a LOD tool, which is a logarithm of odds. And this is one of the ways that we help to identify those families and the genes associated with EDS. Let's see. So, this is as detailed as, this is as scientific as I will get today. I'm not going to talk about each of these different techniques, but each of them have been developed and have made us better in terms of finding where genes are. And with that, we're now looking for the gene that's associated with hypermobile EDS. And the question is it one gene or multiple genes? I strongly believe that it's multiple genes. I'm fascinated by Dr. Maitland's theory and observations about can the hypermobility be a function of an autoimmune disease, and for which then it changes the connective tissue? Or are there connective tissue disorders that are separate from the autoimmune disease in hypermobile EDS? And I think the answer is yes for both of those. I think it's a fascinating idea, fascinating studies that need to be done, and it's very important for us to have put together all these people. I think Charleston and the Medical University will be a collection of researchers and ideas that will bring forward some of these things. I am stuck with being using the 2017 guidelines. For those who don't know, it is a guidelines that has virtually no science to it, in my humble opinion. And I can go over each of the elements of it. I'll just say, for instance, the Beighton hypermobility score was developed not for EDS, but for babies with congenital hip dislocation. Now, how did we get from there into using it for EDS? I understand a little bit about in terms of ease and understanding and doing those sorts of things. You know, where is the science that says that there's an aortic root enlargement? Now, I know that that's in Marfan syndrome, another connective tissue disorder, but where's the data? And there is no data. And so I'm thrilled to see that that the Norris lab has put together a questionnaire, and from that questionnaire we hope to go around the country and use it as a stepping stone to look at the clinical manifestation, compare what patients say and what patients really have, and see if we can get a score that will better identify those patients with hypermobile EDS. So once, you know, again, if we can find the gene or genes, the wave of the future in genetics is, can we find the basic defect? It reminds me of the diabetes story. You know, it's only been in the, you know, the late 30s that we discovered that diabetes was an, you know, was an insulin problem. And now the very first step was we gave animal insulin, and then we started making human insulin. But that's really not, that's treating but not taking care of the problem. Can we figure out what the basic defect is for the connective tissue? Can we give that to the patient? But ultimately, we need to change the gene. And there's, again, a lot of technology out there that's looking at how we make gene changes. And there have been a couple of fascinating studies and some interesting technology that's somewhat scary about making designer babies, but again, hopefully the role that we have is to first treat and then to cure. And with that, I am done. Anybody have any questions? Good. I've bored you enough, I'll get off the table.
Here, no, I have a mic for you. Go ahead.
Okay. I mean, Invitae is a very common genetic testing service that people do once they are pursuing the diagnosis of Ehlers Danlos. And obviously, we don't have, you know, a verified one for hypermobility, but does Invitae tend to cover kind of all the known standardized variants for the other types, or are there ones missing from that test that people should be aware of?
So that's a very difficult question. I would say that each lab picks out a panel of genes that they think are important. I think that they use the genes that are most frequent and most likely to be found. Do they do all the genes that are potentially out there? The answer is no. So I think it is a good screening tool. You know, if you look at the 2017 guidelines, the very first thing that you're supposed to do is eliminate any other connective tissue disorder or Ehlers Danlos. And so that's why I think many of us tend to do a genetic test before we really move on to the clinical diagnosis. We want to make sure that you don't have anything else. You know, I personally have been surprised sometimes in finding things that I would have never thought. I had a young lady who was about maybe 5'2" and come out with a Marfan syndrome mutation. Again, we think of Marfan's as a tall, lanky, and heart defects, and she had none of those, and so it's as good as we get today. Yes, ma'am.
Next question. You mentioned incomplete penetrance. Is there anything associated with sex to that? Because I've heard that males don't often manifest the symptoms of EDS as much as females do. And is that something that you've seen as well? And what's any particular reason for that?
I think that if you're in the EDS world, you probably see 85 to 90% women. And so is that a lack of diagnostic criteria? Is it that males tend to express things differently? Is it a hormonal problem? You'll hear someone who's an expert in neuroendocrine, and maybe she can answer that a little bit better. But I think there is some bias of ascertainment. We tend to see more women than men. One of my pet theories is women produce a hormone called relaxin. Relaxin is a hormone that helps women have babies to come through the birth canal. It opens up the pelvis. You also produce it during your menstrual cycle. Is that, and many women, if you ask, have more problems during their menses than at other times of the month. Again, is that relaxin? Is that progesterone? Is that the variation of estrogen and progesterone? I don't think there's any real research, as best as I know, in really looking at those kinds of issues. Yes, there's someone over there, way over there. So do I.
No, it's sort of anecdotal, but I do have a large family, and we've all kind of diagnosed ourselves with we call it rickety skeleton disorder. And I was wondering, how do you connect with people who might be interested in participating in something that's multi-generational like that?
So I think I think the center that Ann is proposing is going to be a great place for those kinds of studies. Again, I am not well versed in what other people are doing, so I can't say that it's not happening elsewhere, but at least I think it's going to happen here. And again, having all the elements, the gastroenterologist, the neurologist, the orthopod, all being in one area, focusing on one disease like this, or I think will maybe lead to faster identification of the genes and different potential treatments.
Thank you.
Yes, sir. Or yes, ma'am.
So, Dr. Shapiro, I've been wondering, because I've heard some maybe supposition, and I've wondered myself if perhaps this is a copy number variant. Does anyone know a copy number variant gene, and that accounts for why it can be so different between different with the generations? And so I wondered if you had any comments on that idea.
I have not seen any data on that. There are a number of disorders for which copy. For those who don't understand or are not familiar, there are a number of diseases in which you repeat certain sequences of DNA. When you reach a certain point, you start having disease. An example would be Fragile X syndrome or Huntington's disease. Depending on how many copies you have, is whether you're affected or not affected. I don't know that those things have been done. I would think somewhere along someone has looked at it, but I don't have any data on that. I just think that there's such great variability of expression. Why does someone have GI symptoms more than some of the than POTS or and I think that's a function of the. As a human being, we've got about 20 to 30,000 genes. I think it's the interaction of those 20 and 30,000 genes with this abnormal gene that gives us that variation. There's So I think that's, so for those who didn't ask, the question was, is autoimmune diseases being looked at in terms of changing the symptomology? And my answer would be yes. And I think Dr. Ann here is one of those people that are really looking at that avenue. And again, there can be great variability of expression. Nobody has all the signs and symptoms of it, and there must be a reason why, and it's probably the epigenetics of other genes plus the gene that's involved.
Hey, Doc, can you hear questions from the
Hi. What
virtual
exactly
audience?
is the relationship between EDS and POTS? Like, is it causatory? Is it more of a correlation? And why is it only one portion of the EDS population that has that symptom?
So I'll answer the second question first. I think that, again, there's great variability of expression. POTS, really fascinating is that we have seen a marked increase in the diagnosis of POTS with the infection with COVID. So COVID has affected the brain somehow, and some of the individuals with what we would call long COVID now have POTS syndrome. POTS has, could be, from just a purely physiologic theory is a connective tissue disorder. Do you have, do your veins stretch a little bit more, and you collect more blood in your feet, and you have less venous return to your heart and less blood being pumped to your brain, and your brain says, I need that oxygen. The very first thing I'm going to do is I'm going to speed the heart up, and if that doesn't make it enough, the brain says I'm getting gravity out of the way, and so you faint. It happens in normal people. As I tell people, I'm old enough to remember when the Queen used to review her guard, beautiful red coats on a green lawn, and you look down and there'd be somebody flat out. And that individual had a POTS-like episode. He didn't squeeze his legs, didn't get that venous return, and he passed out. So I think that there is, again, that's that variability of expression. Why do people have POTS, and why do people have gastroparesis? And why do people have both? We're not quite sure why that is as of yet, but I think that's an area that hopefully we'll get some more answers on. All right, thank you.